Hi. No. Okay. Hi, everybody. Good morning. While you are sitting, I will have two announcements before we start. The first one is we would like to take a meeting picture just after Gareth's talk downstairs. So please, we should, before the excursion, we should meet all downstairs for a picture. And the second one is don't forget to come back from your excursions to listen to the public lecture of Giles this afternoon. Voilà.
So you have my small presentation. Okay, great. So, in the Mansion of Neuroscience, neuroendocrinology has been, in the eyes of many, a small room, barely a broom cupboard, scarcely worthy of note beside the lounges and ballrooms of cognition. Yet this broom cupboard is where we exercise our passions. It's where we eat and drink, grow fat and hypertensive, make love and war. This text, of course, is written by Gareth Leng, who is here today to give the history of neuroendocrine lecture. It captures the essence of Gareth's creative skills as a writer and speaker in our scientific community spanning many decades. I've worked with Gareth and known him for such a long time, and what always amazes me is his capacity to make all of us think outside of the box and look at some things from a very different angle. My name is Suzanne Dixon. As programme chair, I just couldn't think of anybody better to give this history of neuroendocrinology talk. He's my former PhD supervisor from many years ago, too many maybe, colleague and friend. So Gareth actually started his career as a mathematician. He became a dedicated electrophysiologist when he was doing his PhD, and he was recruited to the Babram Institute, where he was a group leader, was fascinated by pulsatility in neuroendocrine systems. It's there I worked with him. He moved to the University of Edinburgh in the year 1996, where he's been ever since. He's actually very recently moved to France to live. He doesn't really need an introduction to this audience. You know, neuroendocrinology is his middle name, I think. He's a founding member of the BSN and its journal, the Journal of Neuroendocrinology, and he's also held the position of president of the INF, the International Neuroendocrine Federation, previously. So I was so happy to learn there's so many early career scientists attending the meeting, because I was once one a long time ago. And I have some advice for you. You know, you should always listen very hard to the words of your supervisor, because they're very important. So Gareth has given me many words of wisdom as a student. So he would say to me, Suzanne, from a physiological perspective, the hypothalamus is the most important in the organ, in the body, because it controls eating and sex. And by the way, the rest of the brain only exists to support the hypothalamus in this very important task. And another piece of wisdom that he gave me as a student, he said, Suzanne, it's very important. He had heard me practicing a talk, probably for this meeting many years ago. He said, Suzanne, you know, you've got to remember this. When you give a talk, you should always give it in a way that people come back and want to hear what you have to say the next time you're speaking. So, Gareth, we're all here, and the floor is yours, and we welcome you for this lecture.
Okay, well, thank you so much for those kind words, Suzanne. When Suzanne first told me that I was going to be history for this meeting, I thought, fair enough, you know. of course, and then I thought, well, of course, history began yesterday, but then that would be a very short talk. So let me just go back. Facts and Myths, Travels in Neuroendocrinology. That's the title I kind of decided for today. If I can give you the kind of takeaway message, and then you can all go off and have lunch. And I guess when I kind of thought about this, what was the message that I really wanted to communicate? And that was, well, put it like this. You've all been listening to some fabulous talks, really interesting, engaging science, beautiful, convincing narratives. What should you do when you hear a fantastic story? Well, what you should do is look to see where it's wrong. Okay. I think skepticism, above all else, is what kind of characterizes the best science. Don't be seduced by fantastic narratives. Always be questioning. I'm looking forward to the questions after this. I said I could begin history as being sort of starting yesterday, but you have to choose a kind of random date. And 1955 is as good as any. And 1953, I was born in 1953. That was a good year too. And that period was a fantastic period for models. It was a period, of course, when Watson and Crick developed their model of the DNA, Hodgkin-Huxley, the model of the action potential. But of course, here I'm going to begin by talking about what a Frenchman, I'm now French, now I'm a doctored French person, Bruno Larochelle, called the creation myth of neuroendocrinology. And that can date back to Geoffrey Harris, who wrote this fabulous monograph in 1955 called The Neural Control of the Pituitary Gland. And in that is this lovely model diagram. It is a model, it's a very kind of simple explanation of a very complex system. And in that, he proposed the existence, or what he argued provided all the evidence for the existence, of a set of controlling factors in the brain, I know you all know this, that would control the pituitary gland. What you might know is not really be so aware of is how very, very controversial that was at the time. And because when he wrote that book, it was reviewed in Nature, and it was reviewed in Nature by this character. And the review in Nature was subsequently described as the most spiteful and mean-minded review that Nature ever published. And it was written by this character, Sir Zolly Zuckerman. Now, Sir Zolly Zuckerman was the nearest thing that we've ever had in Britain, in this country, to a kind of king of science. He was a phenomenally powerful character. Sir Zolly Zuckerman, later Baron Zuckerman. If God had ever had a kind of chief scientific advisor, it would have been Zolly Zuckerman. As he was, in fact, during the war, he was really the chief scientific advisor to both the British and the American governments. He was a scientific advisor to Allied command. And after the war, he was all-powerful in science, a phenomenally powerful character, really guiding the MRC, chief scientific advisor under successive governments. But why did he take such offense at this? I'm just going to quote some of the key characteristics of his scathing review of this kind of notion. The place taken by the hypothalamus in current biological discussion is in striking contrast to its size. This minute region of the brain also remains a happy hunting ground for experimentalists concerned to find in it neural centers for almost every visceral function of the body. Those with which it has already been endowed are more numerous than are its recognized groups of nuclei, and it's scarcely surprising that some of the latter have had to be invested with the control of very different functions. It's dripping with sarcasm. It ends, Professor Harris might have been better advised if he'd left the problem with a statement. The information as to the details of the mechanism involved is scanty. Instead of proceeding to speculations about the number of chemotransmitters that might be involved, when not even a single one has yet been identified. Now, he, of course, Why was he taken so aggressively about Geoffrey Harris's foundational statement? Well, that's kind of interesting, because although he was a polymath, had enormous control over a vast swathe of science in this country, by his training and his academic lineage, he was a reproductive endocrinologist. That's where he made his name. And this little diagram here of Geoffrey Harris really kind of sketches out really what the challenge was for him, because he, as an endocrinologist, understood the control of the pituitary gland as being purely an endocrinological problem. It was a matter of communication between hormone-producing bodies. And the idea that neuroscientists had anything to say about this was an outrage to him. And so putting the brain at the top of this system really kind of offended deep at the heart of his personality. But of course, you know, as it happened, Geoffrey Harris turned out, and the way of history is he turned out to be right. But not before a very public debate between Zuckerman and Harris, at which the kind of the details of the science behind the different approaches was argued out in public to a crowd that sat agog there. And of course, over the following years, all those chemotransmitters were identified: GnRH, somatostatin, TRH, CRH, dopamine, GHRH. And of course, they won Nobel Prizes for Shelley and Gilliman. Do you know Zuckerman never conceded defeat, not even after the Nobel Prize had been awarded to Gillian and Shelley. He never abandoned his claim that this whole structure was a piece of nonsense. Now, Gilliman and Shelley, of course, engaged in that hunt for these chemotransmitters, partly because they were convinced by Harris's case. I don't know whether they ever read Zuckerman's rebuttal, but they were convinced enough by Harris's case. Now, they won the Nobel Prize, and I think they both conceded that had Harris lived, he died, unfortunately, very young, and had he lived, he would have shared the Nobel Prize. Because they had both conceded before their prize that he was the person who was the kind of founder of that field and that idea. So they were driven to kind of hunt for those chemotransmitters. And why did they get it? Because Harris was in the hunt as well. Well, Gilliman and Shelley won that race, not Harris. Because in order to find these chemotransmitters that Zuckerman thought was impossible to find, ludicrous to imagine, they gathered enough hypothalami to isolate them. Shelley's lab collected the hypothalami from 5 million sheep, 50 tons of hypothalami, to isolate his factors. Both of those labs had huge resources. They did this on an industrial scale. And if you talk to the people, and actually the stories are there, Cy Bowers, one of those involved in the isolation of TRH, said that no intelligent person would be involved in this process. He said that, well, in the time that he did it, he thought his IQ dropped by 20 points. It was brute force. It was brute force following the vision laid down by Geoffrey Harris. But what I want to kind of ask and consider and begin by asking is why, how can you explain Zuckermans resistance? And I think that Zuckermans resistance really comes down to this. He saw that Harris's theory was a kind of existential threat to reproductive endocrinology as it had been. It was taking away, is telling this community of reproductive endocrinologists, of which he was the king, that actually the answer is not with you, it's with the neuroscientist, and this is something he could not live with. That's one version. But another version which we have to kind of reflect on too is that propagated by Thomas Kuhn in one of the books that actually led me into science in the first place, The Structure of Scientific Revolutions. And Thomas Kuhn is probably famous for this kind of statement more than anything else, that scientific paradigms only really change when the old guard who promoted them either retire or die. He thought that people are never convinced They never change their minds. Once they're committed to a theory, they stick to it, come hell or high water, and there was nothing surprising then about Zuckermans Resistance. Now, if that's true, and many people think it is true, that's a challenge because we exist by persuading people. But actually, Thomas Kuhn said, you never really persuade people. Yeah. All you do is provoke resistance. And Harris, of course, then threw the challenge on to neuroscientists. Okay, it's your job now to solve the problems of pituitary and reproduction and all of the other things that come from the neuroendocrine systems. But I think, fortunately, neuroscientists weren't interested. They didn't care. Neuroscientists had their own kind of agenda, and they still have that agenda, which has been unchanged for the kind of 50 years that's followed, or 60, 70 years. I'm losing track. There's the central dogma, what I call the central dogma here of neuroscience. And I'm putting this down here just to contrast it with the emerging vision that you, as part of this kind of community of neuroendocrinologists, actually constructed a vision of the brain that is in profound contrast to the conventional vision that neuroscientists still hold. And I just want to put that in perspective now by pointing out what that central dogma of neuroscience was and is. And it is really that information processing in the brain consists of neurotransmitter release, a small number of neurotransmitters, release at synapses. Everything happens at synapses. Nothing else really matters. And that is tightly coupled to electrical activity. Now, of course, as we as neuroendocrinologists say, yeah, transmitter release has its part, but actually it's peptides. And peptides aren't released at synapses. They're released from all kinds of things. They act at a distance. And there's a huge variety of different model. Part of the dogma of science is that the substrate of this processing, this information processing in the brain, therefore, is the connectome, this map of the physical connectivity. And we as neuroendocrinologists would say, well, it's plastic. Everything's plastic. And it's not just that even the neurons are plastic in what signals they generate. One day they're vasopressin neurons, the next day they're CRF neurons. And their whole targets are plastic, and they're plastic because actually it's not just about what is released. It's about the sensitivity of the target, and that is governed more than anything else by the density of receptor expression. And so if the target can choose and be designed and alter and plastic in what it responds to and when it responds to, then looking at the connectome is kind of an irrelevance, really. And so then the third model is that plasticity of the brain function is just activity-dependent changes in synaptic strength. Well, we've already said, synapses really aren't just a small part of the kind of spectrum of information processing in the brain. And the electrical activity of neurons encodes specific information. Well, who would say that? Who can listen to Dave Grattan and the pleiotropic control of prolactin release, or any neuroendocrine system? We know that multifunctionality is a characteristic of pretty well every neuroendocrine system. There isn't a specified single track of a single function. In fact, there's this, what we are dealing with in the neuroendocrine systems is multifunctional, multisensory cells. And multitasking is at the very heart of everything we do and think about. And the final part of this classic dogma is that actually, if you want to explain intelligence or anything complex, all you've got to do is look at its size and complexity. In a 20 billion neurons, all those connections, you don't need to explain these things. Well, those of us who've kind of listened to and heard the stories of processing in C. elegans and zebrafish and all the kind of wonderful model systems that I've just kind of walked out of the session on, know that actually the idea that simple organisms are not clever and sophisticated, and that these things are not conserved across the whole sweep of evolution is, you just have to look at neuroendocrine systems to see that this is a kind of piece of nonsense. So I think it is a blessing, really, that neuroscientists didn't really want to follow. Because if neuroscience had taken up the challenge, what we do now would be grossly impoverished. Going back to the creation myth, and my compatriot, now that I'm French, Bruno Larochelle, why do I bring him up? He is one of them. He is, I think, the most highly cited scientist working today. You probably haven't heard of him because he's a social scientist. And why am I talking about him? Because we know so much of the insight into Gilliman's work in the Salk and the origins of the neuroendocrinology from a year that he spent there as a student in the lab, as a social scientist. And he described it as an anthropologist amongst scientists. It was an extraordinary, novel way of doing a different kind of science. Because he had trained as an anthropologist. An anthropologist, they go and look at monkeys and their behavior and deduce what they do from that. And he treated scientists in exactly the same way. He didn't try and understand them in what they were trying to do. He just watched them and saw what they did. And he described that, in this, with wit and in this powerful, powerful book. And there were simple things that he saw and simple things of such, I think, deep insight. The first thing that surprised him, he said that, these monkeys, these scientists, all they do is really, they're out, but all they do is publish papers. Papers that hardly anybody reads except the other scientists. And what an extraordinary activity this is, that's all they do. And it seemed that, why are they doing, what is the point of this purpose? And this purpose was to actually either, to make an argument, to strengthen a claim or to weaken somebody else's claim. It is just shifting the kind of balance of credibility in minute ways. And that's why they read them. They read them closely to see how they were cited and how things were said and to look at this kind of balance of credibility. And the whole purpose of their activity was to try and move their claims up from the status of being speculations to the status of being claims, to the status of being plausible, to the status of being facts. And when they got to the facts, they'd won. And so they claimed, being the scientists, that they were merely scientists discovering facts. But he, Latour, the anthropologist, said, no. They were writers and readers in the business of being convinced and in convincing others. And that is the important thing, that when you read a scientific paper, when you hear a talk, that wonderful narrative, you're not hearing an objective account of facts. You're listening to someone who is trying to sell you something. They're trying to persuade you of the truth of something. You hope that they're doing it honestly by objectively portraying facts in a balanced, clear way. But I think you have to ask yourself, is that ever really the case? For Latour, then, as he understood it, what the scientists recognized the value of each paper was to be calculated in how often it was cited, and more than that, in how it was cited, what it was cited for. So, I think if we think about citations, because we all think about citations, we all worry about them, we all worry about how often we're cited, and sometimes we kind of even measure our respect for each other and how often they're cited and how often papers are cited in journal impact factors. And we think there is this myth. There's this myth that of what citations actually tell us. We believe that, in some ways, and it's partly true, it's not a complete bit of nonsense, but in an ideal world, then citations record the debt that one paper owes to its predecessors. You can trace the flow of, you can acknowledge the origins of an idea, you can trace the flow of impact by studying these networks of citation. What you're seeing here is a small citation network that I'll come back to. And if that's the case, then just by studying these citation networks, what we mean by citation networks is the links between papers, what papers cite and how they cite other papers. And what you're seeing on the right here is a citation network. Each of these circles represents a paper. The larger the circle is, it's been cited more often. And you can see from the lines and the connections all the papers that have cited it. That's what a citation network is and what it looks like. And so by studying these, perhaps we can map out the progress of the field, trace the origins of ideas, acknowledge credit, identify credit, and analyze how evidence is brought together to establish facts, to establish a claim. That's a dream. And I'm not saying it's nonsense. But before we go any further, we had better try to understand exactly what it is that citations do tell us. Why do we cite the papers that we cite? That seems to be, if we're ever going to use citation metrics or statistics for anything, we really have to think about that. What are they actually telling us? So how do scientists decide what to cite? And that's been the subject of a kind of a lot of study in fields that maybe we're not familiar with, but there are whole fields of science called scientometrics that study this. In our field, current average references have about 60. That's the average size. And there on the right to one body of neuroendocrinological literature that I'll come to. That's how the average size of reference lists has changed since 1950. We cite more papers. Reference lists are longer. Now, if you think about it, should be fairly obvious to you that the total number of citations equals the total number of references. So actually, papers are being more highly cited now simply because reference lists are a lot bigger. So the first thing you can know is that you can't compare citation rates now with citation rates of papers published 20, 30, 40 years ago, or even 10 years ago. It's just not, there's no comparability. But let's go a bit deeper than that. Why do we, when you have a budget of 60 references, how do you choose those? Well, I'm going to kind of summarize how I think you choose them, and what other people have said you choose them, and you can decide for yourself if it's true or not. When you have your reference list, what you'll see there is you'll maybe see a few historical classics. In my field, it's the Oxytocin field, common one is Dale 1906 as being the kind of the foundation of Oxytocin, which is an interesting reference because actually 1906 was 22 years before Oxytocin was even named. And what is apparent to me is that nobody who's ever cited Dale 1906 as being the origin of Oxytocin has ever read that paper. You won't actually find anything in the paper that approximates to any way that I've ever seen it cited. Well, except my own citations, but that's different. So why do people cite historical classics when they clearly haven't read them? Well, of course, they do it in order to display their scholarship, display their erudition. Okay. It's part of the process of persuasion. You know, you can listen to me because, look, I know all the way back. They will cite some of their own previous papers. Now, I'm not complaining about that. If you don't cite your own papers, how do you expect anybody else to? I mean, of course they cite some of their own papers, and some of them do it extravagantly, but others, really can't have any complaint about people citing some of their own work, but they do. Of course, they cite some methodological papers. I'll come back to a couple of those later. They have to. They're in the methods section. You'll see those. Fair enough. Maybe. They will decide to cite, they will think about who might referee their paper. And they will be sure to include some references to the potential referees. Absolutely sure. Yeah, certain. You'll put those down because actually editors will often look through the references to find referees. And so you will certainly add it. And what you do not want is your paper going to somebody who hasn't been cited. So you'll certainly do that. They will have some what I've called signposts to the totem field. Papers that every cites they will cite just to show that, yeah, I know what I'm talking about. Every cites this thing. These are the big papers. Here they are. And of course, they will cite some papers in high impact factors, journals like Nature. Why do they do that? Because these are great papers? Well, maybe sometimes, but I think more commonly to show people how important their own work is. Look, this is an important field because other papers have been published in Nature about it. So again, these are all subtle kind of nudges, subliminal messages to people that what I'm publishing is important, I know what I'm talking about, and very little to do with actually the origins of any ideas in that paper. They will they have other things. They will sometimes cite early papers into preference to later studies. Now, that's an interesting one. Should you do that? Of course, in one sense, you might say, well, yes, you should acknowledge priority. But it's a fact that most of the first studies in any field are small and unreliable. The effect sizes are higher than normal. Later studies are generally much more rigorous, and they're bigger and more solid, and the effect size is smaller. But actually, the later, better studies are much less cited than the early small studies. It's a fact. And finally, most people will cite a few very recent papers. Why do they do that? You can have your own reason why they might do it. Maybe because they're important or interesting. And maybe just to show that they're up to date with the literature. Because sometimes when you look at those, they seem like a kind of rather random selection of recent papers. Okay, that might sound kind of provocative, but you've got to kind of have a look at these, because one way you can look and test this kind of idea out is to actually look at some papers that have been highly cited. And this is one that, a few years back, Nature published a list of the 100 most highly cited papers, and I looked down those. And one of them caught my eye because it had this title, the Edinburgh Inventory, and I was working in Edinburgh. I thought, gosh, it's been cited 40,000 times, and it's in the Edinburgh Inventory, and I've never heard of it. And so I kind of looked at this just because of that. I was just curious, what is this paper about? And it was the analysis and assessment of handedness. It's a paper that is cited on average now, currently, about 5 times a day. Right? 5 times a day, every day. Still being cited. And I thought, oh, this is an important paper. It's an interesting paper because it was published in 1971, hardly cited at all until 1995 or something like that. So for the first 25 years ago, nobody cared. What you might call a sleeping beauty. It just should give us all kind of confidence. Maybe our paper's never been cited, but maybe in 30 years' time, it'll be cited 5 times a day. We can hope. So I'd read it. I can tell you that it might have been cited 40,000 times. I don't think any of those 40,000 had read it. Because what is this paper? And firstly, it's a very kind of obscure academic analysis of handedness. And it was driven by the idea that you might write with your right hand, but actually sometimes you write with the right hand but throw a ball with the left hand. It's not an absolute thing. There's a balance of dominance, different activity. So instead of calling it left-handed and right-handed, you should have a continuous scale of degree of laterality. And he developed a kind of questionnaire to establish in a very continuous way what the balance of left or right dominance was in any individual. Effectively, it was saying, don't think of left and right-handed. Think of it as a continuum, and we're giving a kind of a way of measuring where you are on the continuum. So how is it used, and why is it cited 5 times a day? Who cites it? Brain images. Whenever you do a brain imaging study, you do it on left-handed people or right-handed people. So how do you know whether left-handed or right-handed? They claim to use this inventory, which, if you remember, was designed as a continuous representation, as a categorical distinction between left-handed and right-handed. But it's slightly worse than that, as was pointed out in 2015, because although they claim to use the Edinburgh Handedness Inventory, very few of them actually do. They all invent their own questionnaires based on rather different principles, based loosely on some questionnaire that will decide whether they're left-handed or right-handed. So why are they citing this? Well, everybody cites it. You might think that this is, I've stumbled across a piece of anomaly. In 2010, Andrea Stang published a caustic critique of the Newcastle Ottawa scale. Now, you may never have heard of the Newcastle Ottawa scale, and you may never have heard of Andrea Stang. Andrea Stang has published 650 papers. He's a bioinformatician. He's an expert in systematic review and meta-analysis. A big, big name in the field. And the Newcastle Ottawa scale is a scale that is used to assess the quality. So if you're doing a meta-analysis, you've got to kind of rate the quality of the studies you're incorporating and weigh them in that way. And the Newcastle Ottawa scale is a way of doing that. And that's what he concluded from it, that actually this scale, which was commonly used then, is a piece of nonsense. He said the use of the score in evidence-based reviews, meta-analysis, can produce highly arbitrary results. It was no question about it. It was a caustic review. And so he was rather surprised to find out how often it was cited, because actually, critical reviews are not cited. So in 2018, he published a paper drawing attention to this, how that it was very widely cited. And he started to see how it was cited. What's it cited for? And actually, it was cited by people who are using that scale as though it was supporting it. These are people, experts in systematic review and meta-analysis. These are the epitomes of rigorous, careful analysis of the literature. And they were citing Stang, who had attacked the useless scale as though he was supporting it, and normally as the only reference in support of that scale. So, and Stang was astonished by it. And he sets it down there. In a random sample of 100 papers citing it, 96 were systematic reviews, the gold standard of rigor. And all but 2 portrayed the commentary as supporting it, their use of the scale, when in fact the opposite was the case. And he says it appears that the vast majority of systematic review authors who cited this commentary did not read it. Well, that was up to 2018. What's happened since? His analysis has just not been noticed. People are still citing his commentary as though it was saying the opposite of what it actually says. So why do scientists cite their papers, and what are they doing? Coming back to it, scientists are really in the business of convincing other scientists. We might hope that citations mean that a paper is good, but papers aren't cited because they're good, but because they're useful to whoever cites it. It adds a piece of ammunition to their case. Now, it'll add ammunition even if they're wrong. It still adds weight to the argument you want to make. And they don't cite, generally, inconvenient evidence. So papers that are confirmatory, papers that say what you expect or want them to say, they're the ones that are most cited. They're the ones most readily accepted. And they're also easy to publish in top journals, because journals like papers that are going to be highly cited. Controversial papers, contradictory papers are tough to get in. And if they do get in, they're unlikely to be cited. For example, and this is work from Gareth Leng, no relation. That's a lie. He's my son. He's a social scientist, and in his PhD, he looked at the origin of what's called the diet-heart hypothesis, which is how did we come to believe that intake of saturated fat was linked to heart disease. And he was studying this in the period up to when the consensus guidelines of the government advice came out in 1984. And he was studying it because actually it's now known that the evidence base before that subtraction was not clear-cut. In fact, often a lot of the evidence was contradictory. And in fact, many people will say that actually that whole advice is misguided now based on what we know since. So how did it come to be accepted? Well, a key part of that case that was highlighted by the summary of the NIH Consensus Conference was that there was evidence that interventions to eat less saturated fat reduced the risk of cardiac disease. Up to that point, there had only been 4 randomized control trials studying that. Of those 4, only 1 was consistent with that recommendation. The other 3 found no benefit of the intervention at all. In fact, one of the 3 thought it had a negative benefit. So there was only one supportive one, and that was the one that's marked here as Oslo. It's marked there as Oslo, but that's my kind of misprint. Was it in a better journal? No, the MRC study was a much bigger study, and that was published in Lancet. The Oslo one was in Acta Medica Scandinavica. So a bigger study that was a negative study in a better-known journal cited much less often. So he asked, why is that positive study cited so highly? He looked at how it was cited, and he looked at how influential people cited it. And he looked at how it was cited in reviews of the literature. And he looked for reviews of the evidence around the diet hypothesis and how the literature was cited. And of course, there were some reviews that opposed it, thought that there was no case for dietary intervention. Those are what we call the critical reviews. And they cited the critical reviews and the neutral reviews, ones that didn't make any comment. They cited the evidence in a really reasonably balanced, sensible way. But what about the reviews that promoted that hypothesis, promoted the guidelines that became standard? There they are. Most of the reviews, of course, were supporting that case, and nearly all of them ignored all of the inconvenient evidence and only cited the single positive evidence. Scientists, Latour's comment, as I said again, scientists aren't in the business of telling the facts as they are. They're in the business of convincing other scientists that what they're saying is right. And that's why I said at the outset, if you hear a seductive story, alarm bells should ring. Might be true. Look for the problems. At around the time that Harris and Zuckerman were engaging in their debate, and I have no problems with that debate, it really followed the logic of Karl Popper, who was a kind of philosopher of science, very influential at that time, still is for many people, very influential. And his thesis really was that good science is hypothesis-driven science. The best experiments are the killer experiments. The examples of great experiments are the experiments that destroy a hypothesis. They don't confirm a hypothesis. He said that actually confirmation has no value unless you put a hypothesis at risk of refutation. Then the evidence has no value. Always, any good experiment should challenge an idea that you think is dogma and attempt to be capable of disproving it. So how we still pay lip service to this commonly. Many people talk about hypothesis basis of their experiments. In 2012, a scientist Daniel Finelli actually looked for papers that explicitly declared their objectives being to test a specified hypothesis. He looked for papers that declared this model of good science as being the model that they were following. He looked at a whole range of fields, and amongst them, nearly 3,000 papers in different aspects of biology. And he reported that of those who reported that they were testing a hypothesis, something like 80% reported they confirmed it. When I looked at that, I thought, that we should pause here. There's something worrying about this. If you make a prediction with an 80% chance of being correct, there's not really any point in doing the experiment, is there? There is only a point in doing experiments when you really don't know, when you're really unsure. So the idea that 80% of your hypotheses are confirmed is just that you're not really being selective about how you're choosing what experiments to do. So confirmatory evidence you really want to look at with some suspicion. And there are lots of explanations for why you get so many positive results. And one of the obvious explanations is that actually some people did the experiments that didn't support their hypothesis, so they changed their hypothesis retrospectively and told the story as though it supported it. One possibility. Another possibility is that actually they did the experiments, and the ones that actually didn't support it, they never got published. They put them in a drawer. They never tried to publish them because they didn't think they'd get published. What we call publication bias. Nonetheless, we've got to be careful of what scientists tell you. And I think you've got to start from the basis that whatever citation counts tell you, they don't tell you anything about the quality of the paper. That doesn't mean to say there's an inverse correlation. It doesn't mean to say that highly cited papers are less likely to be true, although there's some evidence that is the case, especially if they're highly cited from journals like Nature that are known to be less reliable and less reproducible than papers in other journals. But I'm not saying that just because they're highly cited, they're rubbish. What I'm saying is that there's a disconnect between the quality of paper and how often it's been published. And this disconnect in some cases is striking, because actually if you do publish a paper that demolishes a theory, do you expect it to be cited? Why would anybody cite it? Because if it's really demolished a theory and people accept the theory is demolished, they're going to stop working on it. And if nobody's working on it, nobody's there to cite it. And again, you can know examples of exactly that happening. The best examples of good science are almost of nature not going to be cited. So they're not going to, so citation networks and citation counts are going to say nothing about quality. And that extends all the way through. It means that journal impact factors tell you nothing about quality. It means that citations to a scientist will tell you nothing about that. In fact, we know that. Citations to a scientist, I'm a highly cited scientist to some extent. It's a function of my age. It's the fact. Nothing to do with the quality. So, but what do they tell us? Do they tell us anything useful? Well, they do, because they tell you about fashions, and they tell you about trends. They tell you about where the field is moving, whether it's moving for good reasons or bad. You can see where it's going. And the more you look into them, you find that that information is revealing and interesting. And so what we did a couple of years ago is to study the oxytocin field, which is a field that I've worked in for many years, through citation networks. And this network that I showed you before is the beginning of the network oxytocin paper, because this is how oxytocin appeared in the titles of papers and what papers it appeared in the years up to 1960, at the very beginnings of the field. On the right there is a graph of how the number of how often papers have oxytocin in the title over the years to the present day. And you can see that field going kind of up and down over time. What's going on here? Now, to explain what you see before, I've got to explain this diagram here. Those black papers are from the chemists. I remember in the 1950s, Davignon won the Nobel Prize for sequencing of oxytocin and for showing how to synthesize it. And this was the focus of activity there. Those purple papers, these are from the obstetricians who are using the synthesized oxytocin to study the effects in parturition in labor. And in a lot of those use this technique of intranasal oxytocin exposure, because in order to propel childbirth. It was the common way of administering oxytocin in maternity wards through intranasal application. That intranasal application of oxytocin at the time ran into eventually a lot of criticism from obstetricians who thought that the amount of oxytocin that gets in is highly variable by this route. Some patients get homeopathic amounts, some people get excessive amounts, and so it's ultimately a kind of dangerous route for obstetrics. And they also thought that the doses they were giving up to 1 international unit were really excessive. That was kind of ridiculous doses. Just remember that for a moment, 1 international unit intranasally. You might wonder, I'm talking about papers with oxytocin in the title, but lots of papers on oxytocin don't have oxytocin in the title. Well, actually, an awful lot of them do have oxytocin in the title. These are the number of publications with oxytocin as a topic. It's mentioned somewhere in the abstract. Not necessarily a major part of the study. But you can see about half of those who have oxytocin in the abstract or title have oxytocin in the title. Actually, people do put oxytocin in the title. And actually, if you look at the highly cited ones, they've nearly all got oxytocin in the title. But by and large, oxytocin papers are highly cited by other papers that have got oxytocin in the title. So if you want to be highly cited, make sure of that you put something in the title that tells people that it's relevant. Because otherwise, they're not going to know. If you look at how those papers are cited, and how what I've looked here is the papers published in each of those decades, and how often they were cited in the 10 years after they were published. And you'll see how the average citation rate changes. And what you should see is actually that if you look at the size of the reference lists, the number of citations pretty well follows that, not entirely. The citations dropped between 1980 and 1990. And then that, if you look at the publication rate drops then in the following 10 years. Think about it. You publish a paper, then if there aren't so many papers in your field published in the next 10 years, you're not going to have so many citations. So the citations tell you nothing, not about the intrinsic quality of your paper, but it depends on how many other people are working in that field in the next 10 years. This is our network of the oxytocin field. It has about 10,000 papers in it. And that's in the total collection. And what this network is, now I'll explain how these are displayed. Essentially, the algorithm that generates these networks pulls papers together if they have close citation links and pushes them apart if they don't. So what it does, it clusters papers that cite each other. And you can use, knowing that, you can use an algorithm that identifies those clusters based on the interconnectivity and look at those clusters, and they define different communities. And that's how we break up the whole field. And there are in black are the chemistry papers. There in blue are the neuroscience papers. The obstetrics ones are in purple. This orange group, these are reproductive endocrinologists. Well, it's a particular branch, and the green group, these are concentrating on receptors. I'll talk about some of the others maybe. But that's the whole kind of field. But how did that field evolve? Well, I've shown you the beginning of the network. Let's go through it in the next decade. This is 1960 to 1969. And so this is this period when the field is starting to take off. And you can see there's the chemistry group still strong, the obstetrics papers, a few others kind of emerging. What I want to look at here is this graph here, which shows the citation inequality. What you see in this decade is that about 60% of all citations, that's just 20% of the papers. Okay? There's a large inequality. People don't cite equally. Fair enough. Don't expect them to. Okay. If you do the same calculation, same graph for every subsequent decade, these are the same graphs. So these are, they just lie on top of each other. In every decade, papers published in those decades follow the same rule. 60% of the papers are, 60% of the citations go to 20% of the papers. They exactly overlap. It's true in every decade. It's true in every subfield. You break that down to every cluster. It's true in other subfields of science. Citation isn't a kind of, it's a very, very predictable process. It follows what's called a power law. How does that power law come about? It's not what you'd expect if the citation is anything to do with quality, because quality, you expect, they'll give me some good papers published, sometimes some poor years or some weak years. No, every year, every field, same rule. 60% of citations, go to 20% of the papers. So what explains this power law? And the explanation for it is understood is something called cumulative advantage. Basically, papers that are highly cited get even more highly cited. It's been given lots of names. One of the names is the Matthew effect from the Bible. Basically, the rich get richer. So highly cited papers are cited even more because they're highly cited. Why should that be? Well, firstly, because they're more visible. You see the references more often because they're highly cited. Because you see them, you notice them, you put them in your paper. Often, it's because they're written by well-known scientists. So they're given more weight just because the name's attached to them, not the intrinsic merits of them, but merit by attribution. They're in high-impact journals. See the circularity of well-known scientists published in high-impact journals, and they're very visible. Yeah. Do you need any further explanation why they're highly cited? They often, in those journals, they often have lots of authors. Authors cite their own papers. If you've got lots of authors who are publishing a lot, they'll just get a lot, be even more visible. So all of these things are this multiplicative advantage to citations. And then, do you think this is a factor? Reference copying? Do you think actually papers are cited highly just because people copy references without reading them from other papers? Do you believe that? Right. I'm talking about this paper by Van der Geer et al. It's got something like 400 citations. A lot of them in conference papers. But it's 79 times in peer-reviewed journal papers. Rodri looked at these citations, and why he looked at these will be, you'll come to. But he saw that 13 of them were connected together. They're referencing each other. And in each case, this reference is used to support the claim that rutin could dilute the blood, reduce capillary permeability, and lower blood pressure. Fair enough, I haven't told you what the title of that paper is. And those are some of the ways it's cited. That's the title of the paper. Two things about it. Firstly, the title of the paper is The Art of Writing a Scientific Article. How could this be used to kind of support the case for rutin? Well, maybe, you know, maybe it's a miscitation. They meant to reference another point. They put it in the wrong place. Actually, there's something else interesting about the citation, because actually this paper doesn't exist. It doesn't exist at all. The authors don't exist. The title doesn't exist. It was given as an example of the format in which you should format your citations. And it's appeared in 400 publications, including 80 journal papers. These characters, Simkin and Raja, they looked at another way. They looked at reference copying a different way. What they did was they analyzed it when people miscite references, when they cite them wrongly. They looked at errors. We make errors all the time. I've kind of miscited. You get the name of the journal wrong, you get the title wrong, you get the authors wrong, you're on the wrong order. It happens. When you see the same errors occurring again and again, the only way that can happen is really because somebody has taken that kind of citation and copied it. And so they kind of analyzed from the features of how often this is done and how often this occurs. They came to the prediction that actually only about 20% of cited papers have actually been read in the original form. That most references that you see in reference lists are just copied from the reference lists of other papers, of their own papers. And they developed, a few years later, they went this even further. They developed this kind of mathematical model to explain these kind of statistics of citations. And they explain it on the basis of that when a scientist writes a paper, he picks a few random recent papers at random, cites them, and copies some of their references. It fits the kind of statistics scarily well, I can tell you. It's a fun paper to read. I'm not saying it, but it's just challenging. Back to the oxytocin field, where it went in 1779. Here we are. There's the model again. Chemistry paper still there. The obstetrics papers, which are the kind of impact. You think that this is the impact. Oxytocin is an extremely important molecule. It's one of the kind of in the WHO top list of kind of top medicines. If our field has any impact, it is in obstetrics. It saved millions of lives through the prevention of postpartum hemorrhage. They're off on their own. They're disconnected. It's difficult to kind of link them to basic science at all. Just mentioning here one of those papers on intranasal oxytocin, now in 1971, intranasal oxytocin in 1,800 patients, a single study, just how common its use was. Moving on to 1980, 89. Now you see this new cluster. Bang! You see that lovely orange cluster. There are many of you in the oxytocin field there. I bet that I wonder if any of you might guess what that cluster is about, because it's now disappeared in history. Because that cluster was actually all from studies in sheep and cattle on ovarian oxytocin production. It's a source of oxytocin that we don't even think of as neuroendocrinologists. Forgotten. It's lost in history. Fantastic studies at the time. We see new fields emerging. We see that yellow cluster, which is the first appearance of behavior, 1990. This is the time when the citations are dropping. And as I said, it's two questions here. I've explained why the citations are dropping. They're dropping because there are fewer papers to cite these. But why is the number of papers dropping? Big increase in now the field is the dominant factor in the field are receptor papers, different community altogether. But why are publications declining? Well, actually, it wasn't just the oxytocin field that was falling in that time. In fact, most of neuroendocrinology is falling there. This is the number of papers published each year with oxytocin or vasopressin or prolactin, sorry, Dave, or a luteinizing hormone in the title. They're all collapsing in this time. Why are they all collapsing? Because they're collapsing, of course, the citations in those fields are all also collapsing at the same time. Why are they collapsing at the same time? It actually is, back in the 70s and 80s, been the appearance of radiomyoassays. A new technique swept into the field. All of the fields were in about measuring these in the plasma. Those studies in those years were employed radiomyoassays extensively. People came in, did all the easy stuff, picked the low-hanging fruit, and then things got a lot harder to do, and the whole thing fell out of fashion. Not because they'd answered all the questions, but because the questions that were left were actually too hard. Big studies stopped doing it. 2009, here we are. The papers, the field's just beginning to recover, and there's this little red field. What the heck is that red field? It's just appeared for the first time. In the following decade, wow, it's a tumor. Okay. This red cluster is a new field using intranasal oxytocin. This is a community that think they've invented it. It's a new technique, never done before. They haven't even noticed that it was used 50 years ago in huge numbers of studies. They're now giving it at doses of 40 to 80 international units intranasally. Remember, the obstetricians thought that 1 international unit was going over the top because of its peripheral actions. This new community doesn't believe it has any peripheral actions. They think it's a way of getting it into the brain. They start by promoting it as a way of selectively targeting the brain. Fine, what can I say about it? I've said it all. This was the lead paper that I showed you. The data is from 5 decades. There's one exception, which is the decade that followed this. And this is the lodestone of the field. Oxytocin increases trust in humans. It's been cited more than 2,000 times. It's an absolute outlier. It's been an outlier, although it's failed replication by many independent studies. Eventually, the senior author decided what he'd do is he'd do a pre-registered replication study himself of that paper and didn't replicate it. They come out by saying our pre-registered analysis show that the simple and general hypothesis that oxytocin increases trust can no longer be endorsed. Okay, it'll be interesting to see if it has any impact at all on the citation rate. Anyway, we have a review out in Philosophical Transactions just now looking at the evidence behind oxytocin as a social peptide. Through our network analysis, you can break down the sources of the evidence behind it, and you can look at the strength of the evidence by studying each of those clusters separately. You can make your own up of mind and read it. The conclusion we came to from it is if the most highly cited papers in most segments of the evidence represent the best evidence, then the case is weak. And I can tell you that that was a pretty understatement. Okay, I'll come to an end. Suzanne's there. I'll just mention that I'm not trying to sell anything, though the book is very kind of respectably priced. I'm just showing you here one of my reviews, which. Actually, one of the things about Harris is that review from Zuckerman, he had it framed on his lab wall. I loved that. Donovan, his student, relates that in his autobiography. He framed that caustic review. Anyway, that's one of the reviews that we had, and that's from my son Arno, my little blah, blah,